This is Flexo, a tissue engineering company based out of Hillsborough, North Carolina. This product enables you to grow cells in a flexible membrane inside the wells of the culture plate shown on the right. Using a vacuum controlled by the FX5000's computer system, you are able to apply stress to the cells during culturing and observe their response.
After several discussions with the client, we determined the basic need of the client. Flexo grows bats, or bioartificial tendons, between the two anchor tabs. A picture of a bat is shown on the left. A picture of the Flexo culture plate well is shown on the middle image. And notice the tabs where the bats attach. They have been experiencing higher compaction rates and therefore pull off with the bats. Pull off is when the bat contracts so much that it pulls completely off the anchor tab. And a picture of pull off is shown on the left. When the bat pulls off, they are no longer able to analyze the bat. They wish to examine other anchor materials in an attempt to fix this problem.
The goal of our product is to analyze different products, including the foams we ordered from United Foam and the Cerex Flexo currently uses as anchors, to determine which material have qualities that produce the best bats. These anchors produce bats with lower compaction rates than average, a lower pull-off percentage, and the material should operate well within the FX5000 tension system. A die cutter was used to cut the foam sheets into the anchor pieces used in the culture plates. Here's a video of cutting anchor using the die cutter.
Materials can be toxic to cells, a quality called cytotoxicity. Before we began making bats, we had developed a test to determine which materials we ordered would not be toxic to the cells. Live cells appear long and pointy, similar to the shape of an eye. The picture of live cells is shown on the left. Dead cells appear circular, as shown on the right. In the test for biocompatibility, a specific weight of each material was soaked in a medium of methanol and water. The medium was agitated to promote toxin extraction. Cells were cultured in a 96 well plate using different concentrations of the toxic medium. After several hours, the cells were observed under a microscope and given a rating from 0 to 4 based on the ratio of dead cells to live cells. 4 equals all live cells, but 0 equals all dead cells present. This graph shows the results of the biocompatibility test. X's indicate the cells were dead, but probably due to contamination because the higher concentrations are fine. Therefore, these wells are ignored.
The foam was inserted into a 15 milliliter test tube filled with 10 milliliters of dissolved water. The foam was degassed using a centrifuge. After centrifugation, for 10 minutes, the water level after degassing was recorded. This difference is the volume of water displaced, or the volume of foam material. The difference between the geometric volume and the volume of the material is the volume of the pores in the foam. From this value, you can calculate porosity percentage shown on the right for each material.
As shown in the picture, a trough loader is placed underneath the culture plate, and when a vacuum is applied, the bat hydrogel is poured into the trough created. After allowing the gel to culture for 2 hours, the vacuum is removed and the bat is formed. This video shows the Flexo chemist performing this process. We were able to produce bats for only 4 different culture plates due to cell supply shortage.
We put the bats created in the FX5000 tension machine and performed an intensive strain regimen. We first started with the strain rate of 0.1 millimeters per second for 24 seconds. Then we preformed a dynamic sine wave with amplitude of 0.5 millimeters for 3 minutes. This is a video of the FX5000 failure regimen, performed on bats from each material type.
ScanFlex is a scanning program that takes a specified amount of scans of the bats in specified time intervals. The ScanFlex scanner is shown here in the incubator. The Zyflex software uses the ScanFlex scans in contrast to determine the area of the bat. By converting pixels to area, Zyflex can calculate the change in area of the bat over time.
The instrument is used to test the strength of bats and to develop stress strain curves for the materials. This is a video of one of the bats being pulled apart on the instrument by a 250 gram load cell. The stress strain data from the instrument is shown in this graph.
To conclude, we were able to grow and test bats using 4 different anchor materials, and the bats from each of these culture plates were acceptable, with an average compaction rate and very low pull-off percentage. We also made testing protocols for the biocompatibility test and the porosity test. In addition, we have created a method for performing tests on anchor materials in determining suitable angle materials.
Luke and Davis done a great job.